Hey guys, I hope you're all doing well, and we're into May now, which is exciting. It's our last month of school. Here's Lily, say hi. All right, we're going to talk about DNA today. So we're into chapter 10, which is our last chapter of the year, so that's really exciting. And we're kind of keeping on our topic with genetics, but going a little farther into the molecule that actually gives us our genes, which is the DNA. So in 1928, this guy named Frederick Griffith, who is from Britain, he studied a bacterium called Streptococcus pneumonia, which causes pneumonia in mammals. And he was trying to develop a vaccine because people were dying from it, and that's really bad. So the disease-causing strain is called a virulent strain of a bacterium. So he injected a live S strain, which is one that is surrounded by polysaccharides, and this capsule just keeps that disease safe from your immune system. So the R strain is going to be one that lacks this capsule. So it is still disease-causing, but because it doesn't have that capsule, your immune system can actually attack it. And he was doing all these experiments with mice. So here he was. He had 4 different sets. Here's the R strain, which the mouse survived because it was able to attack those disease-causing cells. Here's the S strain, which ended up killing the mouse. He also tried a heat-killed S strain. So it was still the bad strain, but because it was killed, it actually kept the mouse alive, which is kind of how we make vaccinations, one little way. And then he also did one with a live R strain and a heat-killed S strain, and this one actually killed the mouse. So he wasn't sure why the mouse died this time, because he knew that the R strain shouldn't have hurt the mouse, because whenever he did the regular R strain back here, the mouse survived. So he couldn't figure out what exactly happened. And this is just basically going through and telling you everything I just said. So at first he found the S strain killed the mice, R strain kept it alive. Later on he heat killed the S strain and R strains. He found that the mice survived the heat killed S strain, but was dead after the S strain was mixed with a live R strain. And here are the words in case you need that. So non-virulent was the R strain, virulent was the S strain. When we heat kill it, mouse lives. When we heat kill the smooth strain but mix it with a live R strain, the mouse dies. He concluded that the heat-killed virulent bacteria is going to be released by a hereditary factor, and that's going to transfer the disease-causing ability to the live, harmless R cells. So essentially, the dead S strain is transforming the R strain to be really bad. And he called this a transformation. In 1940, Oswald Avery's goal was to prove if DNA, RNA, or protein was a transforming agent. So he knew there was 3 things that were in cells, and he wasn't sure which one was going to be the one that ended up killing the cell. So he's using an enzyme called protease, which is going to destroy protein. RNase is going to destroy RNA, and we have DNase, which is going to destroy DNA. So they found that the cells missing protein and RNA were able to transform R cells into S cells and kill the mice. But the cells that missed the DNA, so after he killed the DNA off, those cells couldn't transform anything and the mice continued to live. So the conclusion was that DNA is responsible for the transformation in bacteria, and you guys already know that DNA is what has all of your genes on it. So here is a picture example. We have the protease, which is killing the protein. The mouse is going to die. Lipase, which is just breaking lipids down, inject that, the mouse dies. RNase, if we inject a mouse with that, it's going to die. DNase, if we inject a mouse with that, it will keep it alive. So DNA is the transforming principle here. Well, the Hershey-Chase experiment in 1952 came about to see if DNA or protein was the hereditary material viruses transfer when they enter a bacterium. So they're specifically talking about bacteria, they're not really talking about us right now. So viruses infect bacteria, they're called bacteriophages, and we can shorten that up to just a phage. We like to shorten everything in biology. So here's an example of what a phage looks like. It has DNA up here in this little gem thing, and then it has these little probes, and these little probes, or its legs, are what actually go and they connect to the cell, and that allows it to inject its DNA. So they're using a radioactive isotope to label the protein and the DNA phage. So they're trying to light it up so they know where the DNA is. They allow the protein and the DNA to infect E. coli. and then they remove the phage from the cells. So after the phage injects its DNA, they take the phage off of that cell. And they used a blender to do this. They spun it around really fast and it just flew off. They used a centrifuge to separate the phage from the E. coli. Centrifuge is basically the same thing as a blender. It was spinning things around, splitting things apart. They found that all of the viral DNA and a little of the protein had entered the E. coli. The conclusion was that DNA is the hereditary molecule in viruses as well. So not only do we have DNA, but viruses have DNA. So here we got the little virus. This rectangle is the cell. It's going to attack the cell and it's going to inject its DNA into the cell, which is actually how viruses work right now. So then your cell ends up recreating that viral DNA. All right, we're going to stop there for today. Hope you guys have a nice rest of your week.